Hello, my name is Jeff Brabec. I am a fourth-year graduate student at the University of Vermont, and today I'm going to talk to you about a method that we have built to perform an integrative multi-network functional prioritization in Alzheimer's disease to identify targetable genes and pathways in early AD pathology. Thanks, and hope you enjoy. Recent large AD meta-GWAS have identified up to 38 genome-wide significant loci. However, GWAS hits have yet to yield effective therapeutic targets for the disease. While these significant loci explain much of the disease risk, they don't account for all the dysfunction observed in AD pathology. Methods like network-based gene reprioritization, or NGR, lend functional context through tissue-specific functional networks, but for complex diseases like AD, there are multiple relevant networks ranging from cell type to the whole brain, with no clear best choice. We hypothesized that a multi-network approach to NGR would yield the most conservative functional predictions for AD genes. Finally, we performed an integrative analysis of NGR functional score, genes involved in AD disease subnetworks, and early pathology single-cell differential expression data to identify candidate genes and pathways with the best therapeutic potential. We chose to work with the three brain cell type networks, as these are the lowest functional unit of brain tissue and where the genetic and molecular dysfunction manifests. We processed our data through two parallel pipelines. First, the three cell type networks were dimension reduced and merged together, then processed through our NGR pipeline to get disease gene candidates. For the single network analysis, each individual network was processed through our NGR pipeline to yield disease gene candidates. Linear SVM is standard for NGR, but we believed that for this multi-network approach, a nonlinear method might perform better, so we chose to use a Gaussian SVM. Finally, the functional scores were integrated with the single-cell RNA-seq p-values, and pathway analysis was performed. In NGR, you take a disease-relevant tissue-specific functional network. For AD, you could use the hippocampus, and you note which genes in that network reach a nominal level of significance in your GWAS, noted here in orange, and which don't, noted here in gray. NGR works on the idea that nominally significant GWAS genes live in functional networks with other GWAS genes and genes whose regulation they might alter, noted here in red. Therefore, we can use a machine learning algorithm to classify genes as functionally associated with the disease, or not, by the number and strength of their connections to the nominally significant GWAS genes. We can then use the resulting functional score to re-rank our genes. We performed our NGR using nested cross-validation, and for our training data, we observed that the multi-network performed as well as the single network examples, though better than the neuron network alone. We picked the best models from our training set and applied them to our holdout testing set, and observed that for the Gaussian SVM, all models performed equally well, though the multi-network model had less variance. For the linear SVM models, however, the multi-network significantly outperformed the single network examples. These results support our hypothesis that the multi-network approach to NGR is more conservative at predicting AD candidates than the single network approach. Next, we wanted to examine which of our functional candidates also had differential expression in early AD pathology. To do this, we used the joint cumulative distribution function. The joint CDF takes two or more criteria and calculates a score based on a gene's performance on both axes. So this gene in yellow would have the best score because it does better than 7 out of 10 genes on both criteria, whereas this gene only does better than one other gene on both criteria, so it would get a score of 1. That analysis yields plots like this, where the points are colored on a gradient from highest score, noted in yellow, to lowest score, noted in blue. We performed the analysis with the astrocyte and excitatory neuron differential expression data. For the neuron, we observed genes like ERP29, PSMD7, and COPS6, which are localized to the endoplasmic reticulum and are all involved in ubiquitination pathways. We also observed SOD1, which codes for superoxide dismutase, which helps combat oxidative stress. For the astrocyte, we observed genes like GFAP, PLEC, and CSF1, which are all involved in glial response to lesions. Pathway analysis in the excitatory neuron integrative ranking revealed among many others, pathways involved in the regulation of cell death, as well as neuronal synaptic plasticity. These results replicate a previous analysis of genes involved in AD-specific neurodegeneration performed earlier this year by our lab. Some of the pathways enriched in the astrocyte integrative ranking are involved in gliogenesis, stem cell proliferation regulation, as well as the regulation of locomotion. It seems that at this stage of AD pathology, the astrocytes are beginning to respond to AD-related neuronal death. Based on previous observations in our work, we hypothesized that at this early stage of AD pathology, neurons are dying due to excitotoxicity-induced oxidative stress, causing lesions in the brain, which trigger astrocytic production and movement in an attempt to heal the injured tissue. We believe that this makes the astrocyte a strong candidate for therapeutic intervention at this early stage of AD pathology. With that, I would like to thank my lab group, my dissertation committee, as well as our collaborators at Jackson Labs for making this work possible.